Panel discussion and to go to Dr. Chinmaya Mahapatra, who is a pharmacologist and pharmacist. He will present the mechanism underlying the COVID-19 with the current scenario of vaccines, and he will speak about the safety for sure about the vaccine. He's a pharmacovigilance associate in the Indian Pharmacopoeia Commission in Ghaziabad, which is belonging to the Ministry of Health and Family Welfare, Government of India. He has created and is a very successful editor-in-chief of the Journal of Pharmacovigilance and Drug Research with very high-level articles, and one of the founding members of the Global Pharmacovigilance Society. Even if it's a very young society, he has already organized several conferences, international conferences with people from Latin America and Europe and US and Africa. So thank you, Dr. Chinmaya, to start your presentation, and very, very glad to welcome you.
Thank you, Dr. Erin. Thank you so much for inviting me to present my talk. At the outset, I would like to thank all the persons who have joined in this event, and thank you for their time. And I would like to thank Dr. Peter sir and Dr. Pipasa ma'am, and that's it. And a very good morning, good afternoon, and good evening to all of you. And few my friends are also there in this event. So let's go to my presentation. I will be talking about the mechanism of coronavirus and how the vaccine acts in our body. So brief about the manufacturing process of the vaccines, and after that I will cover the things like the mutations, whether the vaccines are effective or not. So shall I share my slide right now? Hello?
Yes, I think that you can share your screen.
Yeah, just a minute.
Are you allowed to share your screen?
Yes.
Okay,
Thank
great.
You. Is it okay now?
Yes, great. No, you have to do it.
Yeah,
right.
sure.
Okay, now it's good.
Thank you. So, in this presentation, we would be knowing about what is COVID vaccine, how it works, how it is getting manufactured, and whether it will protect us from this pandemic or not. Is this the only solution, or any other solutions are there? Whether it causes any adverse events or not, whether the vaccines are safe, then whether the mutation in coronavirus is harmful to us or not. So my presentation will cover all these things. So going to the next slide. So the COVID-19, as we all know, the term was coined by WHO on February 11, 2020. It indicates, the CO indicates corona, VI indicates virus, then D indicates disease, and 19 indicates 2019. So now coming to understanding of the COVID-19. Let's say we have taken the vaccine at first dose we have taken, so we have to wait for 4 to 6 weeks and to take the second dose. Then after taking the second dose, we have to wait for 2 weeks more. So it is around 45 days we have to wait, so that our body gets totally immunized, means our immunity system will get boosted up, and we have to wait till 45 days minimum. Okay, so if at that case, if the vaccine fails, like as Dr. Maya Sharma described about the efficacy and all, so if the vaccine fails, then we may get affected by the virus. Okay, so we don't know the incubation period. It is not yet been confirmed that how many days it will take to get the virus infected by infect, and the symptoms, the first symptom that come. But taking into the account the first symptom, that is mild to moderate fever. Mild to moderate fever is normal, because as there is a reaction, then there will be an equal and opposite reaction, right? So we will get suffer from any kind of mild to moderate things. It may be a sore throat or sneezing or something, something we will get. So that will be our first day. So we have to note it down. Then from as the day passes on, if we won't take any medicine at that time, then we will suffer from all the other kind of symptoms like dry cough, then loss of taste and smell. So up to 4 to 5 days. So in between the day 1 to 5, if we take the medicine with consultation with the physician, then we may get cured. And we have seen many patients are being cured in these 5 days only, if they take the medicine properly. But if they delay by taking, some person means around 15 to 20%, they don't believe, don't want to go to physician, and they take the drug by their own, the OTC drugs they take. So it prolongs the disease, means they don't know, means whether it is we cannot identify the things like. So if it passes on, then from 6 day onwards, they will start suffering from breathing problem, or something they will start. So at that time they will reach to the hospital, and they will say, sir, I'm suffering from such kind of things. So I think I have got suffered from COVID. So can I go for COVID test? So I think at that time, if after 7 to 8 days or 9 days also, if someone approaches a hospital and if he goes for a RT-PCR test, then the test will come as negative. What I have observed, it is just only my point of view. It is not any other things. So I'm just explaining it just little bit. So the drop in oxygen level is not due to coronavirus. It is due to the uh, antibody what we have in our body, and they destroys our internal lumen of the blood vessels in that veins. So they ruptures the internal lumen of the blood vessels, and they start forming a clot. And at that time the patient won't get oxygen properly, and he felt the less in breathing. So the complication starts. Then if he won't get proper treatment, then it will go for some, if will prolong for some day, then the fever again reappears, and then he suffers from the pneumonia. Then at that time it will be very late, means the stage is very difficult to handle at that time. So but if as per means, as I have seen, many patients are getting recovered with proper treatment. So the identification of the first day is very important to treat the COVID 19 thing. And the vaccines are just a barrier for us, so that we won't get into that infection. So moving to the next slide. So there are different types of coronavirus, like human coronavirus, bat coronavirus, then feline coronavirus, canine coronavirus, then porcine coronavirus, alpha coronavirus, and beta coronavirus. Now coming to the types of human coronavirus, there are 7 types of human coronavirus. So 229E, OC43, NL63, and HUK1, and SARS-CoV in 2002. The generally the people are thinking that the virus is appeared just now, means last year it appeared and we are suffering, but not like that. Earlier it was there, and in 2012 it appeared as middle east respiratory symptom it caused. So the first 4 variety they produce common cold, so that is not a issue. But in last stage, means in 2019, the virus what appeared, it is a genetic cousin. That's why we named it as a SARS-CoV-2. So it is not a new one, it is a genetic cousin of the earlier one, and it is producing more serious infections. Now coming to the structure of SARS-CoV-2, so it is having its in the inside it is having positive single standard RNA, and which is enveloped by enveloped glycoprotein, then membrane glycoprotein, then the nucleocapsid proteins and all, then the spike protein, spike glycoprotein. So now if we take a close look of spike glycoprotein, then it has two subunits, S1 and S2, and the S1 is the main thing which is responsible to bind to our uh, cells receptor. Okay, so coronavirus is an envelope spherical structure about 120 nanometer in size. You can imagine how much small it is. So now, in fact, how it enters into a host cell. So as most of us will know, we know that it binds to SE2 receptor, and it enters into the cell by cell fusion. So now, after entering into the cell, it starts multiplication. It starts its transcription process, then its translation process. It produces proteins, and it gets assembled again, and it comes out from the cell, and and then we will feel we can't speak, we can't feel the taste also. So these kind of things, it will show as a symptom. So now at this point, the vaccine plays a very important role. The vaccine, what means what we are developing, it is just producing the immunity. That I will explain in my next slides. So so now coming to the S protein, it is having uh, 1273 amino acids. Okay, so single peptide, that is divided into NTD, that is N terminal domain, then receptor binding domain. It is the very important thing, and the S2 subunit is divided into fusion peptide, then heptapeptide repeat sequence, then TM domain and the cytoplasm domain. So now, if we are listening that some mutation has occurred, whether it will be effective or not. But mutation is a natural process. We cannot stop the mutation, but and the mutation, it can occur in any region of this chain. Okay, so if it occurs in any part, then there is no need to be worry. But if it occurs in RBD, that is receptor binding domain, then we have to think of it, means whether it will be means we have to it is a variant of concern, what we can say. So so as per earlier studies, the mutation that occurred at D614G plus I472V, it is the most crucial one, and as per the study, means it increases the infectivity and decreases the sensitivity to neutralizing monoclonal antibodies and the convalescent sera. So it is the only means as per their study, whatever they collected the data, as per their study, it is the most deadliest mutant. Okay, so now mutation is not a old one. The D614G, it has appeared in the year 2020 in China. Then in cluster 5, that mutant, it appeared in Denmark in August and September 2020. Then in December 2020, the SARS-CoV-2, the variant of concern, the 2020-12/01, it appeared in UK, and later on in the December 18, the 501Y, that V2, that appeared in South Africa, and now in India, the double mutant strain B1.617 also appeared. Uh, Apart from that, E248 484Q. So the E248Q is a indigenous strain of India, and the L452R strain is from California. So if these two viruses um, come together, then they forms a double mutation. So this is what the mutation we can't stop, but we can try our best to um, stop these viruses. So how our immune system works, we have our body to fight with the coronavirus, so no need to be worried about that. But the macrophages, whatever are there in our blood circulation, so they digest the germs and dead dying cells, but few cells they get remained, and they are termed as antigens, and they start stimulating our body to produce B lymphocyte and T lymphocytes. Now if B lymphocyte they produce, then they produce the antibodies, and which kills the antigens. Okay. Now the T lymphocytes, normal cells, means the normal cells infected by the antigens, they are getting killed by T lymphocytes. Okay. That's why the consequences of the cases are very severe in some patients. But the T lymphocyte, we have to the vaccines are we have to develop more T lymphocytes, which are being stored as a memory cell for us, and they will fight back with the virus when we may get affected in the future. So now coming to the how COVID-19 vaccine works. So after taking the vaccine, it takes few weeks to produce huge amount of memory T lymphocytes and B lymphocytes. But there is a chance of infection if not taken care until the vaccine doses it work. Means that 45 days, what I had showed it previously. So we have to cover up the 45 days very carefully. Then only we can we can feel safe. But still we are not safe, because the efficacy of a vaccine, it varies from one person to another person. We cannot give a particular number that it is 80, 70, 60, like that. We can't give a number. In practical situation, it totally it varies. Okay. Now type of vaccines. So there are three types of vaccines, like mRNA vaccine, protein subunit vaccine, and vector vaccine. Apart from that, there is another vaccine, that is DNA vaccine. So all the things are having are same mechanism, like they produce some harmless protein in our body, and our body develops antibodies to eliminate that proteins out from our body, and few are remaining as a memory cells. And whenever the next we will get affected by virus, then the virus will produce the proteins, and at that time they are going, they will go and they will attack that things, and they will kill that virus itself. So mechanism was all the same, but the thing is different. Means the parts whatever they are used during the manufacturing of the vaccine, it differs. Okay. So in mRNA vaccine, they use the mRNA of uh, okay, it will come. So vaccines that are available right now, it is mRNA vaccine is Pfizer and BioNTech vaccine, then Moderna vaccines, then protein subunit vaccine, that is Epivac Corona, then RBD dimer, then vector vaccines like Sputnik V, Oxford AstraZeneca vaccine, COVID Convidecia, then Johnson and Johnson vaccine, and the another form of vaccine also there, like inactivated vaccine, what we use, that Covaxin, the that we are using here. So these are the other forms of vaccines where live virus is inactivated during the process of manufacturing the vaccine. Now coming to the Pfizer biotech vaccine, that is Moderna vaccine. So they prepared lipid nanoparticles by taking five constituents: ionizable cationic lipid, then neutral lipid, then cholesterol and pegylated lipid, then mRNA. But the thing is that here the main challenge is the storage condition. So as in a cold chain, if it exposes to a larger temperature, then it may get destroyed on the transport itself. So there is a means that is the just challenge in this vaccine. But the other things are good. The technology used in a Covishield and Covaxin, these two vaccines what we use in India. So Covishield is a viral vector based technology that was used for manufacturing Ebola vaccine, and the Covaxin was prepared by whole virion inactivated coronavirus vaccine. made by the same technology used for influenza, rabies, and hepatitis B vaccine. Okay, the composition varies. The In Covaxin, the composition they included the inactive coronavirus, that is NIV 2020 770 strain. Now, if some new strain appears and if the vaccine won't work or its efficacy uh, uh, is uh, is not there for that strain, then we can't say that whether he means we can't predict something like this is good or this is good, like that we can't predict because the virus is changing its strain in a natural process, right? Now, composition the of Covishield, it is inactivated adenovirus with segment of coronavirus, then aluminum hydroxyl gel, histidine hydrochloride monohydrate, then magnesium chloride hexahydrate, polysorbate 80, then ethanol, sucrose, sodium chloride, and disodium edited dehydrate. So all these things they took and they prepared these then they prepared the concentrate of the vaccines. Now, what happens once they prepare the concentrate, they have to prepare it in a large scale, in a production scale, right? So in the there are 5 steps in manufacturing COVID vaccine in a industry. So what they do, they first clean clean the stopper, cap, and the bottles, vials, everything they clean and they sterilize it. Okay, that is the first step. Then the second step, in the formulation tank, they vaccine concentrates are diluted with solvent to make specified volume. Then in the third stage, they have the filling units, so where the fully automatic isolator for filling of vials and in a aseptic area. Okay, and in the fourth stage there will be one quality check, that is for clarity, then particulate matters, then assay, then it will go for packaging and dispatch. So this is the manufacturing process. So it depends on the capacity of the industry, how much they can produce per day. Okay, now coming to the reporting of adverse event, we have adverse event following immunization system in India, AEFI reporting form. So till date, on 20,586 reports have been submitted to the um, through the AEFI system, and the data has been taken from COVID app. So that's it. Then the ADRs associated with COVID vaccine. So if we open our laptop and we visit the VG based software, VG Access software, then we can find that the adverse drug reactions are multiple in number, but in total, if you type Covishield or Covaxin or Pfizer, Moderna, whatever you type, it will show the number of ADRs related to the corona vaccine only. So it may be anyone, it may be any one of the vaccines given. Okay, so it is showing 78,070, sorry, 7,80,073. Okay, ADRs. Out of that, 5 lakh ADRs are there because of the general disorder and administration site condition. And if you go inside it, then you can find it out that number of deaths are 5027 totally ADRs reported. So the number of persons has been taken the doses. It is 1.45 billion vaccines they have has been given. So now if we calculate the percentage of death, then it comes to 0.00035%. So as compared to the mortality rate, or by comparing somewhat, we can say that the vaccine is safe, and we should take the vaccine, and we should protect ourselves to go inside the to go into the incubation period of the virus. Okay, so that I can say here. And this the 522760, that is here it is a generic disorder, so it is very less, means ADRs related to any kind of COVID vaccines are very less. So we can say that as a statistically we can say that it is quite safe to take the vaccine. Now coming to the fact sheets, that is for Covishield and Covaxin. Now it has got updated. Okay, yesterday only the things came that we can give it for 12 days, 12 week, the second dose. So it is got changed little bit. Now coming to the adverse events, the here in India around 80% persons has taken Covishield vaccine, and after taking the vaccine for 30 minutes, they are staying in observation period, and meanwhile, at that time, they feel some kind of head reeling, then vomiting, then myalgia, then just minor symptoms they are getting, the injection site pain, like they are getting. But later on, after 6 to 8 hours, someone some are returning to us and saying, some we are suffering from these things. So we cannot there is no such data to correlate the probability or possibility, whatever the scale we use in WHO UMC or Naranja scale we have for the casualty assessment. But we can say that yes, we can note it down, and we can send it to our regulatory body, and they have to decide whether it is related to the drug or not. That is their work, and they will do that work. So the thing is like that, means few persons have got affected by demyelinating disorder. If you look into that, what is that disease? That is a disease in any condition that results in damage to the protective covering, that is myelin sheath, that surrounds the nerve fiber in our brain, and optic nerves and spinal cords. So when the myelin sheath is damaged, the nerve impulse slow and even stops. It causes neurological problems. So yeah, then this is the Covishield. And now coming to the Covaxin, same thing. There are injection site pain, then headache, fatigue, then fever, body ache, then abdominal pain. So all things are most almost similar. So there is no such major difference between these two vaccines at ADRs. But the thing is that some signals it will may come later on, means it when we will give it to 18 to 34 years of age group. That depends. Now coming to the conclusion, so I think it is my opinion that the COVID vaccine can protect a human, but there is no 100% efficacy. Okay, so COVID is curable if identified at the early stage, that is day 1 or day 2, where the patient feels minor fever and myalgia. Person should promptly go for RT-PCR test on the day 1 or day 2 itself, and if he feels uneasy, then the symptoms of the dry cough starts from 3 to 4, day 3 and 4. Better to start the therapy with physician's consultation instead of going for OTC drugs or dry cough syrups. They won't work at that time. Then the oxygen support requires in serious conditions from day 5 to 6 onwards, with all the therapies prescribed by the physician. Then patients should follow all the instructions given by the physicians to get the recover quickly. So 14 days battle can save the life. That is my last concluding remark. And I would like to thank all the person who has given their valuable time to listen to this talk. And thank you, Dr. Irene, for organizing this event with a lots of effort. I can understand because I'm also organizing the event, so I can understand what you have done. So now, if any question is there, I'm ready to take that one.
Hello.
Yeah.
Hi, it's Dr. Hirschman. Just a quick question. Could you summarize the mechanism whereby the mRNA vaccine causes the production of B and T lymphocytes?
Mechanism of mRNA vaccine, right, sir?
Correct.
Okay. So the mRNA, that is a mRNA vaccine, right? Whenever it enters into the, whenever we administer into our human being, okay, it replicates it. It goes for replication, and it produces a protein finally. Okay, it produces a protein, and to destroy that protein or to destroy that antigen, our antibody will work, right?
Yes.
So our antibody will recognize that this kind of protein are there. If it is coming, then we have to destroy that, right? That is for
Right.
vaccines. That is for vaccines. Now it has been got stored as a memory cell in our body. We have not yet infected by the viruses. Now, after few days, if we will get infected by the virus, let's say virus entered into our body and it produced the same protein. Okay, after multiplication and all, it produced the same protein. Then then what happens, our memory cells, that are T lymphocytes and B lymphocytes, they will become active and they will kill that protein itself, if it is going to that stage. Okay, but meanwhile some drugs are also there which stops the multiplication of the virus at the multiplication stage, or many things are there, sir. So
My question goes back a little further. When once you have an mRNA vaccine, you introduce it into the body. What is it about that mRNA vaccine that produces the protein? Is it the mRNA itself that produces it, that causes the cells to produce, or does it have something to do with the mRNA causing its own immune response that then produces the proteins that incite the production of B and T lymphocytes?
I can help.
Yes.
Yeah.
Go on. Who can answer? I wanted to.
No, as you wish, Irene.
No, please, Cyril.
Yeah, so basically, the basic idea is that the mRNA, which encodes only a part of the spike protein, is uptaken by cells in our body. Mainly, I would say APCs like dendritic cells, macrophages, etc. And those cells will read the information encoded in the mRNA the same way, we produce our proteins. So this is a recipe to make this part of the spike. And then those cells will produce that protein or that fragment of protein. And there you will have two concomitant processes. First process, some of that protein will be expressed on the surface of those APCs, of those antigen-presenting cells. And partly, some of that protein will be degraded in small peptides that will be presented also on the surface of these cells to T cells. Now, T cells will recognize that, will be activated by fragments of that protein, and then B cells will come and talk to those macrophages, recognizing that viral protein. T cells will activate them, and then the B cells will produce antibodies. So that's more or less a general scheme.
Okay, fine. That's good. Appreciate it. Thank you.
Perhaps we can say that, in fact, there is really two different passes for the vaccines. There is one a usual pass that we are giving to the body the antigen itself, in a way that it's an inactivated virus or a dead virus, or we are giving, this is a new technologies, we are giving the recipe for the body to in terms of mRNA or DNA, so that we are able to fabricate ourselves the spike protein without being in contact with the virus itself. This is a two big direction of the vaccine manufacturing. Do we have other question?
I have a question. Dr. Chinmaya,
Yes.
thank you for your presentation. And you win for best Zoom background, so congratulations. My question is on pharmacovigilance. Two related questions. The first is, have you seen any blood clotting issues? That's my first question. And my second question is, have the adverse events come from urban areas or from villages? Thank you.
Second question, sir. Can you repeat the second question, sir?
Yes, the second question is, the adverse events that have been reported, have they largely come from the urban centers, from the cities, or from villages?
Okay. Sir, we have, first of all, I would like to answer the second question. Sir, we have ADR monitoring systems, ADR monitoring centers around 346 centers are there across the India. So the patients who are taking the vaccines, if they are reporting it to us, then we are reporting it um, to the regulators, right? So if they are not reporting it to us, then the person who is administering the vaccine to them, he is reporting it to the regulator through another channel, right? So anyhow it is getting recorded, sir. Means in old cities also, in small city, in small villages, in big cities, anywhere the patient is taking the vaccine, and if he suffers from a ADR, then we are noticing that one, and we are recording that one, sir.
Yes,
Okay.
I understand.
Now,
But my question is,
first,
from a percentage perspective, are they largely coming from cities or from villages?
percentage, sir, I think I have no idea on that, sir. So I should not say anything about that. But now coming to the first question, sir, whatever you were asked, that clots, blood clots. Yes, the blood clot is not related to the coronavirus, right? So it is our immune system, our antibodies, which destroys the internal lumen of the blood vessels, and they start forming a clot, right? So they make
No,
the surface rough,
we are
and
speaking about vaccines, not about the COVID. The blood clots that have been, the thrombosis with thrombopenia that have been described
sir, there is no sufficient data to say that yes, it is due to the COVID. That there is no sufficient data, sir. We have to generate more data, then we can say that yes, it is due to this vaccine or that vaccine, we can say. But there is till date, sir, there is no sufficient.
So there's no evidence in India that any of the vaccines have resulted in blood clots.
Sir,
I just want to clarify. So are you saying that there's no evidence in India that blood clots have been caused by vaccines?
yes, evidence is there, sir, for minor, means I think few cases have appeared, but
yeah,
sir,
Dr. Chinmaya, if I could add to what Dr.
yes, sir,
Peters,
yes, sir, you can answer,
yeah.
sir. Yes, sir, you can
So,
answer.
Dr. Peters, I think what you're asking is the causality. So, causality, we have reports of thrombosis, which has been reported in the Indian patients. But the Indian government, whatever reports is available in the public domain, has denied that these are related to the vaccine. So, the causality analysis, which was performed by the AEFI, Adverse Event Following Immunization Committee in the country, has gone ahead and said that these are not related to the vaccine, and especially Covishield is the one, the AstraZeneca vaccine, which is in focus. Yeah, so to answer your question straight, I would say data is there, but causality analysis has not proved that it is related.
Thank you.
This is Pipasha from UK, and there were reports of sudden deaths in India soon after the vaccinations. Have then those reports been analyzed, because those are serious incidences that needed to be analyzed to, of course, causality and other confounders, but were those ones analyzed?
I think that is a great observation, and yes, I think what maybe Dr. Chinmaya would be able to add more because he is with the central government. But whatever we have been told, that these have been analyzed, and the government still believes that they are not related with respect to what the causality analysis was performed. So that is what is there in the public domain.
Okay, so do you think then that because there is not enough good quality information, it is difficult for causality analysis?
Ma'am,
well, I can, yeah.
Ma'am, we have to prove it statistically significance, ma'am. We can't mean if 5 billion people are taking, ma'am, 5 billion people are taking vaccine, and we are getting 10 cases of death. Then we have to see the benefit risk benefit analysis.
Absolutely. There is no question about benefit risk here, because the benefit far outweighs the risk in this condition of when you are treating COVID. That is, there is no argument there. What I'm trying to say is, because we wanted to look into the cluster of cases where there has been reported cases of sudden deaths, whether there have been a complete analysis, and those would be qualitative analysis of those data. But do you think, because of very poor quality data that you guys are receiving in India, you cannot do the assessments, evaluation of those causality properly?
No, ma'am. As per India, the quality of the data is very good, ma'am. As compared to other countries, we stand at number 9. As compared to
I'm
other
not talking about other things. I'm talking about vaccine now. Because
can I add something here, Dr. Pipasha? So this is Arshia. So I have also been following up and looking at the data, and I've reported quite a few cases to PVPI, where there were a few cases where I saw there were sudden deaths or a breakthrough infection where I wouldn't say breakthrough, because only after first dose of the Covishield vaccination, there were cases where the patient experienced or died within 15 days. But then he was also COVID positive, and he had comorbidities of diabetes. And so I had reported all that information, and we expect that PVPI and the government officials are taking into account all the data that is coming in, and then assessing the causality correctly, and then like the AstraZeneca BRAC report that came in from EMA, that also had 8 cases of thrombosis that were reported from India, and there was causality assessment done there as well. And of course, causality assessment really depends on the kind of data that is coming in, the kind of, if the patients have reported the comorbidities or not, the concomitant medications.
That's what I said.
Exactly.
Because of the poor quality of data.
So if you, I think the COVIN app actually, which I'm going to talk about in my next presentation, does ensure that when the patient is vaccinated, I mean that when the healthy people are vaccinated, then they definitely are observed for the next 30 minutes or so. And then of course, as Dr. Chinmaya is mentioning, that you are counseled when you receive the vaccine that you must report back any ADR which you're facing to the nearest center. And the government recently has changed that particular timeline. It was initially 14 days. Now it has been changed to 28 days because they believe there could be delayed adverse events as well. So I think, poor quality data is an issue across the globe, and India will not be an exception. But wherever the vaccine is being administered, you have adequate monitoring. Post that reporting, yes, that's a challenge because we are not making direct calls to the people who are actually receiving the vaccine, or they don't have access to, I would say. But the COVIN app, and that is again the part of the presentation I'm making, allows you to report your adverse event. So it allows you to report your adverse event just as the yellow card in the UK. So that is
and also the fact sheets for, and also the fact sheets for, if I can say here, fact sheets for Covaxin and Covishield have the information where they expect that people are going to report the events as well. PVPI has given, and
no
also
one is going to voluntarily report the events.
I understand. Yes,
that is the problem. The problem is there has to be a sincere effort in following up those cases, because you have the patient data. Patient particulars to go and ask those questions, because currently the AFI reporting rate with COVID vaccines in India is woefully very low. It is 0.014%, which is given about how many 40.7 million people have been vaccinated fully. We would have expected a better data. So that's what I think.
It's very true, ma'am, and it's really concerning. And I personally have been trying to, spread information as much as possible so that this happens. I would also like to add that I am a part of a volunteer group in India where we are working for COVID patients, and there is an effort underway where we are going to make people aware of, when the vaccine dispersal is done, or encouraging people to report side effects. And the volunteer group that I'm a part of, so we are calling COVID positive patients at the moment to ensure that they are getting the right kind of support in the in terms of when they require hospitalization, the kind of drugs, the doctor consultations, etc. And the effort is underway to bring this knowledge to people, to make them aware about reporting of side effects as well. And probably, since I'm working on this, I will also make an effort that if it is possible, if we can also solicit this information from the patients, from people who are receiving the vaccine, so that the data is augmented and there is more information on the side effects of the vaccines. And a very valid point, it has to increase, the reporting has to increase.
I think India has, I have to leave. So yes. Thank you for a great presentation, and thank you, Irene, for organizing this. Unfortunately, I'll not be able to stay back for Shubhra's presentation, but I'll touch base with him. Thank you so much. Bye.
Thank you. I would like to add what we are doing in Israel. Everyone who has received the vaccine has received an SMS asking for reporting adverse event. So I don't know if it's efficient, but at least we have a lot of data that can support some analysis. But we will go to that at the next session. I want to first of all thank very much Dr. Chinmaya for his great presentation, and I would like to introduce the next speaker. Dr. Subhrajyoti Bhaumik is a doctor, a gynecologist, and pharmacologist, and he's a clinician. Please,